In this section, we'll discuss the branched chain amino acids. The branched chain amino acids are leucine, isoleucine, and valine. They get their name because they have branching of the hydrocarbon chains on their side chains. These are all essential amino acids, have to be obtained in the diet. Their common degradation pathway takes place in mitochondria and has 3 steps. One is amino transfer, that is, getting rid of the amino group. There's then an oxidative decarboxylation and a modified beta oxidation. So this is a schematic diagram describing those 3 steps. The first is the deamination. Then there's the decarboxylation. And then there's a modified beta oxidation in the 3rd step. And those 3 steps are in common for all 3 of these amino acids. Now the end point of leucine is the production of HMG-CoA, and this is a precursor of ketone bodies. So because of that, leucine is one of the 2 amino acids that is purely ketogenic, that is not gluconeogenic at all. Isoleucine and valine both produce propenyl-CoA, which is a 3-carbon acyl chain. Odd-chain fatty acyl-CoAs like propenyl-CoA can't be beta oxidized. Instead, they have to be converted to succinyl-CoA, which is a TCA cycle intermediate. And these reactions require cofactors, both biotin and vitamin B12. And here's the process by which this occurs. Propenyl-CoA is converted by propenyl-CoA carboxylase to methylmalonyl-CoA. Methylmalonyl-CoA then is converted by methylmalonyl-CoA mutase through 2 steps to succinyl-CoA. Now, propenyl-CoA carboxylase, like all carboxylases, requires biotin as a cofactor. Methylmalonyl-CoA mutase requires adenosylcobalamin, which is a modified form of vitamin B12, which is depicted in this picture on the left. The endpoint of this is succinyl-CoA, which can then enter the TCA cycle. And for this reason, both isoleucine and valine are gluconeogenic amino acids. Now, these processes can be defective, as indicated in the following clinical vignette. A 4-day-old girl is brought to her doctor after suffering a seizure. Her mother reports that she's been vomiting for the last 24 hours and seems less responsive. A physical exam reveals an obtunded infant who's hyporeflexive with increased muscle tone. Her urine has a peculiar sweet odor. Labs show hypoglycemia and ketoacidosis. This patient has a disease called maple syrup urine disease. Maple syrup urine disease is an autosomal recessive disorder caused by deficiency in the 2-ketoacid dehydrogenase complex, which is responsible for the oxidative decarboxylation of all branched chain amino acids. And so this enzyme is in common in all 3 of these pathways. This defect causes the accumulation of branched chain amino acids and ketoacids. And this causes problems by several mechanisms. One, the ketoacids are toxic to tissues, as are the branched chain amino acids in excess, so it causes tissue damage and cell death. In addition, there's an acidosis which causes dysfunction of many cell processes, just like any acidosis would. So this is a metabolic acidosis. In addition, the accumulation of branched chain amino acids inhibits the transport of other amino acids into cells, so it causes secondary amino acid deficiencies as well. The symptoms occur shortly after birth and include vomiting, seizures, coma, and death. Treated patients may have varying degrees of mental retardation. The maple syrup urine odor results from the excretion of these ketoacids in the urine that have sort of the sweet smell to them. The treatment consists of a diet which is low in branched chain amino acids, preventing the accumulation of these toxic metabolites. Now, there's a couple other diseases that result from defects in the pathways of branched chain amino acid metabolism. One is propionic acidemia. This disease is due to a mutation in propenyl-CoA carboxylase. This is the enzyme, if you'll remember, which converts propenyl-CoA to methylmalonyl-CoA. The symptoms of this disease are seen in neonates, and they consist of a severe metabolic acidosis, resulting in dehydration, lethargy, and coma because of the accumulation of propionic acid. The ketoacid comes from 2 different sources. One is elevated levels of propionic acid. The other is inhibition of the citric acid cycle by propenyl-CoA. This drives acetyl-CoA to the formation of ketone bodies instead of going into the citric acid cycle. The treatment consists of dietary restriction of branched-chain amino acids, similar to what we saw in maple syrup urine disease. In this case, biotin supplementation may help because it will maximize whatever residual activity is there. One other disease is methylmalonic acidemia. This disease is due to a mutation in methylmalonyl-CoA mutase. This is the enzyme responsible for converting methylmalonyl-CoA to succinyl-CoA in the branched-chain amino acid degradation pathway. This is, remember, is the one that requires vitamin B12. So this reaction may also be inhibited by a deficiency in vitamin B12 due to a defect in the enzyme which produces the particular adenosyl cobalamin. This disease is also a disease seen in infancy, again characterized by severe metabolic acidosis, by accumulation of methylmalonic acid and propionic acid. There's also hypoglycemia, which is secondary to inhibition of pyruvate carboxylase and gluconeogenesis by methylmalonyl-CoA. Again, the treatment is the same as in the others we've talked about, dietary restriction of branched-chain amino acids and high-dose vitamin B12 to maximize residual activity. Next, in the same section, we want to talk about the degradation of a few other amino acids. And these amino acids are lumped into a category that we call one-carbon metabolism. Now, we call it that because of this feature of biosynthetic pathways. Certain compounds require that there are single carbon groups get transferred from one molecule to another as we're degrading some processes, some macromolecules, and synthesizing others. And this process is carried out by molecules which carry these single carbon groups from one molecule to another. And these are things like tetrahydrofolate and S-adenosylmethionine. There are also donors which give up these one carbon groups and recipients which incorporate the one carbon groups to form these new compounds. So here's an example of how this might happen. Here are some donors on the left: choline and methionine, serine, glycine, formaldehyde, histidine, and formate all can donate single carbons in various oxidation states to all of these things: phosphatidylcholine, proteins, nucleic acids, creatine, serine, glycine, thymine, purines, etc. Now, one of these carriers is one called tetrahydrofolate. Tetrahydrofolate is derived from dietary folate. We can't make it. We have to consume it in our diet. And it carries donated methyl groups bonded to the N5 or N10 nitrogens, shown here. Here's an example of a donation reaction to tetrahydrofolate. Serine, in the degradation of serine, serine can donate a methylene group to tetrahydrofolate, and that leaves glycine, and then this tetrahydrofolate containing a methylene group. The glycine then can be further catabolized to carbon dioxide and ammonium while donating another methylene group to tetrahydrofolate. So in this process, we can completely degrade serine to glycine and then to carbon dioxide and ammonia, completely degrading those amino acids. Now, this is also reversible. We can use it to take ammonium ion, carbon dioxide, and methyl tetrahydrofolate to form glycine, and glycine then can be converted to serine. So it's a reversible process. Another donor is histidine. In the catabolic pathway of histidine, there's a donation of a methynyl group to tetrahydrofolate. The result is the formation of glutamate in the degradation of histidine. Now, once tetrahydrofolate has accepted these single carbon groups, it can then donate them to form other things. We've already talked about how tetrahydrofolate can donate single carbon groups to synthesize glycine and serine. We'll also see that it's involved in thymine and purine metabolism, donating the single carbon group to the nitrogenous bases of DNA and RNA. The other thing you can do is to donate single carbon groups to S-adenosylmethionine, which is another one carbon carrier. S-adenosylmethionine is an important donor of methyl groups in the formation of a whole bunch of different compounds, including phosphatidylcholine, in epinephrine and catecholamine degradation, in creatine formation, in methylating proteins and forming nucleic acids, in synthesizing melatonin and other things. So S-adenosylmethionine is formed and used in a cycle which includes homocysteine and methionine. So here we talk about the metabolism of another amino acid, which is methionine. So methionine is formed from homocysteine by the donation of a single carbon group. That's added to homocysteine to form methionine. Then a sugar group is added, and a adenylyl group is added to form S-adenosylmethionine. S-adenosylmethionine then will donate the methyl group that it got from tetrahydrofolate to form all of the other compounds that we've talked about already. In the process, it's converted back to S-adenosylmethionine. It loses its adenosine and is converted back to homocysteine. And this cycle can occur over and over and over again to form S-adenosylmethionine and then to donate those single carbon groups to other things which need to be made. Now, how are these products degraded? How do we get rid of homocysteine? Well, this really goes to the heart of how we metabolize methionine. If we have too much dietary methionine, we need to get rid of it in some form or fashion. We use the cycle shown in the previous slide to convert it to homocysteine. Then homocysteine can be combined with serine to form cystathionine, and cystathionine can be broken down to alpha-ketobutyrate, which is an intermediate of the TCA cycle, and the amino acid cysteine. So in this process, we've not only degraded methionine, but we've also formed cysteine. So this is both a degradation as well as a biosynthetic pathway. There's a defect in this pathway, which is illustrated by the following clinical vignette. A 13-year-old girl is referred to the pediatric clinic because of recurrent deep venous thrombosis. This is an unusual finding in someone this young. A review of her medical history reviews mild mental retardation with a few episodes of psychosis. She's legally blind due to dislocation of the lens of her eye. Physical exam is significant for kyphoscoliosis, which is abnormal curvature of the spine, and pectus excavatum, which is a defect in the formation of the sternum. She's tall and thin and has extraordinarily long fingers. Laboratory tests are negative for anything that would indicate that she's hypercoagulable, that she has a tendency to form clots. But the plasma methionine and homocysteine are elevated. This patient has a disease called homocystinuria. Homocystinuria is an autosomal recessive disease caused by a mutation in cystathionine beta synthase. This is the enzyme that's responsible for the metabolism of homocysteine to cystathionine in combination with serine. Pathologic features of the disease are those that we've described in the vignette: ocular malformations, particularly lens dislocation. They have musculoskeletal abnormalities, including tall stature, long fingers, pectus excavatum, and kyphoscoliosis. They have central nervous system defects, usually mild mental retardation, and may have episodic psychosis. And the most important thing is they have vascular manifestations. Homocysteine is toxic to vascular endothelium at high levels, and this irritates the endothelium and leads to the formation of blood clots, causing recurrent thromboembolism. All of these manifestations are due to the accumulation of homocysteine. The first-line treatment of this disease is large doses of vitamin B6. Vitamin B6 or pyridoxine is a cofactor for cystathionine beta synthase, and 50% of patients will respond adequately to high doses of pyridoxine. For the other 50%, you have to give them a protein which is low in methionine. So this is the end of the lecture on brand